These secondary structures and tertiary structures, the three-dimensional shapes, there are certain substructures within protein structures that have special functions, which we call them, and which we observe these small shapes in many proteins. I mean, the shape may be different, but there may be certain substructure, certain subshape, which is observed in, for example, 100 different protein structures. These are observed and they may serve for a special function, like this helix loop helix motif. These are called motifs, structural motifs. And this helix loop helix motif is observed, for example, in proteins that bind DNA. Okay? Certain proteins, they interact with the DNA directly. And the helix loop helix motif is central. It plays an important role in this binding. So if you have a protein structure that have this helix loop helix motif, it may be you may predict that it's a DNA binding protein. So certain substructures may give you clues about function. This 4-helix bundle, or usually there are proteins, membrane-embedded proteins, for example, with 7 helices. Okay, so they also have, they may all be membrane-based, but their specific function, a protein have a general function like that. When you first see a protein, you may first generally say something about that protein. For example, saying that this protein is going to be a membrane localized protein. That's something general. Okay, but there are lots of membrane localized proteins. What does it specifically do? For example, it can detect sugar, for example. That's going to be its specific function. Specific, in addition to these helices that is going to help its localized in membrane, it may have certain small regions, a small loop region. A small loop region may have some amino acids that are very important to determine its final specific function. Okay. So first I'm going to start the first problem we are going to talk about is secondary structure prediction, but I'm not going to spend too much time on this problem because the techniques here, I'm going to talk about really old techniques which are based on certain statistics about amino acids. For example, each amino acid has a certain preference, usually amino acid types, they have certain preferences to be in certain secondary structure. Some amino acids, they prefer to be in helices. Some amino acids, they want to be in beta strands. So by looking at, if we have a large structural database, by observing these statistics, which amino acids take part in which secondary structure. By looking at this observation, then what we can do is most of these statistics-based techniques is they go over this sequence and they slide a window. Okay, we may have a window of size 10, for example. In this window, just look at the preferences of each amino acid within this window and sum it up. For example, all of them, if all of them prefers to be in a helix, so it may be a high probability that this window is part of a helix structure. So statistics-based techniques are like that. They slide a window, and by looking at the preference of each amino acid in that window, they accumulate these preferences. And by using certain static rules, saying that if the preference of alpha helix is more than this value, then it's predicted to be a helix. If they prefer to beta sheet, so that by using certain fixed rules, these methods, by going over this sequence, they predict the secondary structure. And predict the secondary structure is also one-dimensional information. It's still an array of characters. And this is like this sequence, these 5 amino acids are going to be part of beta strand. These amino acids are going to be a helix. There's probably this is probably a wrong prediction because usually we do not have 2 amino acids forming a helix. It's a very short helix. Probably that's an incorrect prediction. So what we can do is after we predict based on this window sliding, we can also do additional corrections by using some additional domain knowledge. For example, if you know that to be a helix, you need to have at least 4 amino acids, for example, because you need a turn, and the first amino acid interacts with the 5th amino acid in this turn, so you need at least 5 amino acids. So if you detect a region in which is predicted to be a helix and it's short, then you can say that it's probably not helix. We did something wrong here when doing this prediction. So secondary structure is an easier problem than 3-dimensional position prediction. So that's probably, and most of the tools use it as a first phase to predict 3-dimensional structure. So before finding out where each amino acid is going to be in 3-dimensional space, the first logical step would be first identify which part is going to be a helix, which part is going to be a beta sheet. Because after this first level, what you have is you have a higher, instead of individual amino acids, you can work with these secondary structure elements when determining the overall 3D shape. For example, if you identify the beta strands by using a secondary structure prediction tool, then the problem becomes which beta strands interact with each other to form beta sheets. You reduce your problem solution space tremendously by considering secondary structure elements instead of amino acids. So that can be used for these purposes. Secondary structure prediction is important. There are statistical methods, this Chow-Fasman and Gore. There are other neural network-based predictors, CyPred, JPred, PhD, support vector machine learning-based techniques. So there are numerous techniques that predict secondary structures, and overall their accuracies are around 80%. Helices are easier to predict than beta sheets. So if a protein contains mostly helices, these tools may achieve accuracies in 90% accuracy. They may find which amino acid is going to be part of which secondary structure. If your protein is composed of mainly beta sheets, they are less accurate. Most techniques are, it's more difficult to find beta strands compared to helices. And what is the main reason for that? The main reason is that the beta sheet is a result of an interaction that are of amino acids that are far from each other in this primary structure, in this sequence. Helix is just a local, it's really a local structure. By just looking, consider this window sliding technique. A helix is just going to be a helix by itself. It's not going to interact with other amino acids that are far away in this structure. It's really a local structure, a helix. However, a beta strand, to make it a strand that's straight shape, it needs the interacting other strands to form this sheet. And this strand may be very far away in the sequence. And that makes prediction of beta sheets really difficult. I mean, by just looking at this local window, you may not be really sure. You need its interacting partner, which may be very far away in this sequence, to actually be confident in your prediction that, okay, these are the two beta strands, they interact with each other. So it requires some kind of 3-dimensional information to be able to accurately predict beta sheets. Therefore, their prediction is more difficult compared to helices. So there are also assumptions that are made by some of these techniques. And one is assumption, I mean, this assumption is also for 3-dimensional structure prediction. They also use this. And the entire information for forming the structure is contained in the sequence. So what does this tell us? This tells us that you should be able to, by given an amino acid sequence, you should be able to tell its structure by just looking at its sequence. There are no other factors than this sequence itself that determines the structure. Okay? This assumption is actually not 100% true, because There are some helper proteins, chaperone proteins, for example, which help. So there are external factors in the cell that determines the final shape of the protein. So the protein structure itself is not solely based on its sequence. Based on external factors, where this protein is going to be localized. If a membrane localized protein may have a different shape than, the hydrophobicity of the physiochemical properties of amino acids may mean different things if it's a cytoplasm localized protein or a membrane localized protein. So environmental factors also affect the final shape of the protein. And most of the techniques, either three-dimensional structure prediction techniques or secondary structure prediction techniques, most of them ignore this fact. They just say that this is the sequence, I'm going to predict the structure based only on this information. Okay, so this is a big assumption. And other assumptions are for secondary structure prediction, for example, they make these additional assumptions like examining a window of 13 or 17 residues is sufficient to predict the secondary structure. This 13, 17 amino acids, by looking at them, you should be able to tell what the secondary structure is. This is their assumption, and they usually slide a window of that size. However, again, this assumption is not true, especially for beta sheets. As I told you, beta sheets are results of long-range interactions within the sequence. So this is called long-range interaction, because a subsequence interacts in 3D space. So this is why it's called long range. So yeah, actually we have some beta sheets here. You see these regions here, these cylinders at the end of this table. Let's assume these are your beta strands. If they interact with each other like this is what I have here is an interaction. They bind each other. This interaction is called a long range interaction because in sequence they are far from each other. One of them is at one end, the other is at the other end. We have a long-range interaction here. In 3D space, since an interaction means that they're going to be close to each other in 3D space. So this kind of an information violates the third assumption, the long-range interactions. Therefore, it's difficult to predict those beta sheets. I'm not going to, I already told you the general idea behind these techniques. They slide a window, they look at amino acid preferences in this window. Each amino acid has a certain preference to be in a certain secondary structure. They accumulate these preferences to a single number, and they look at the single number to predict what that window is going to be. Okay, so they have some fixed rules like that. If this preferences is greater than this number, this floating point number, then predict it to be a turn, for example. So they have this fixed set of rules, fixed logic, which uses these preferences, accumulated preferences, to predict what that amino acid is going to be take part in. Gore method is similar to, it's also a statistical based method. And this figure, I just put it there to show you to give you a general idea about the accuracies of secondary structure prediction methods. As you can see, the accuracies lie on like 80%. So 80% of the sequence can be predicted correctly in most of the cases. But for certain things, certain methods, so the best method seems to be this light gray type of, which is, I think, the Cypred. Cypred is obtain high accuracies, and these are different tests. And as you can see, Cypred does really good to predict helices. This Q3H shows the accuracy in helix, and Q3E, E is the acronym for extended beta strand. They use this single letter E to indicate beta sheets, and its accuracy drops from 80% to around 73%. So it's more difficult to predict in general these. And these are the coils turns. Turns are also named the parts that are different that are not helices or beta strands are called coils, and they're also predicted over 75%. So the accuracies are around 75 to 80% in this. This was a paper that was published in 2004, so fairly recently. It can be considered recently. And this is again a motivation slide that protein structure is really important. If you know protein structures, this is another visualization of a protein structure. Now the coloring is different. It doesn't, the red region and blue region are not the chains anymore. This is again, we see a surface representation of this protein, and the colors have a different meaning. It shows the electrostatic potential. Okay, it shows charge regions. For example, this red region here may be a pocket that bind the compound. Okay, it shows which region of this protein can be candidate pockets that drugs can bind to. So this again just to show you there are very, very different ways of visualizing protein structures. Structure and function are really related to each other. By just so it's really important to say something about the final function of a protein. And structure-based drug design makes use of the structures of proteins to design small molecules like this. So this is a small molecule, small compound, which is designed. Okay, that's hence the name drug design. So you say, let's take these two atoms together, form this chemical structure, this chemical substance compound, and what this one was is HIV protease inhibitor. Okay, this is the name of this drug. It inhibits this protein, HIV protease. HIV protease is a protein of the HIV virus that is synthesized by this virus. And by inhibiting this, you just kill that protein of HIV virus. But the HIV virus is so, it's so robust. I mean, even if this protease is killed, it can use other ways to function. I mean, even if the protease is killed, it can use other proteins to basically infect the patient. I talked about experimental techniques here. The main things are X-ray crystallography and NMR. I'm going to skip those. And X-ray crystallography, they really need really large experimental facilities, like this is electron accelerator, I guess, here. And we need to crystallize the proteins. NMR is a different technique which tries to predict the structure in soluble form. So the main advantage is we can find the native structures of proteins in their native environments in solution. But the disadvantage of NMR is that it cannot solve large molecules. It's limited to small molecules. It cannot find large protein molecules. And what it provides is these both X-ray crystallography and NMR, they cannot directly give us the atomic coordinates. They are indirect techniques. In other words, the X-ray data, you use some computational algorithms, some tools, to convert the X-ray data you have, to convert it to coordinates. You are solving a puzzle. Like you are seeing some picture of a certain protein from a certain view. What you're trying to find out is that, okay, I'm seeing this picture. What can be the coordinates of these atoms so that they produce this picture? Okay, so even with this experimental techniques, it's not very trivial to find the final coordinates, but the techniques are very well established. They can give you the coordinates. For NMR, it's a more difficult problem to find the coordinates, because NMR just gives you, the NMR data at the end gives you a list of constraints, saying that, for example, this atom and this atom cannot be far away than this distance. This atom and this atom cannot be far away than this distance. So it gives some distance constraints, it gives some angle constraints. It gives you a list of constraints like this, the NMR technique. And your goal is to come up with a certain set of coordinates which satisfy these constraints. So it's like an optimization problem here. It's very difficult. And at the end, a multitude of different coordinate values may satisfy these constraints. So an NMR structure usually gives you, an NMR gives you at the end a set of structures, not just a single coordinate list. It may give you many, many different coordinates for a single atom, but usually certain regions, the constraints in a certain region are more strict. It's going to give you more certain coordinates. You cannot see it right now, but you can look at on the slides later. The end regions of these proteins, for example, since we have less constraints, the coordinates are very variable. I mean, we have different coordinates for end residues here, but for certain regions here, we have more constraints and more strict fixed coordinates for atoms in the middle here. Now, I'm going to talk about our second problem is how can we find the three-dimensional shape of a protein given the amino acid sequence. So this is the sequence for the protein lysozyme. And this, given the sequence, can you predict that its shape is going to be this? Okay, how can we do this? And it's called the protein folding problem. And it was listed among the top unsolved millennium problems in the beginning of 2000s. I mean, this is one of the still unsolved problems of the century. You may have seen, like in the news, what was the name of the project? I don't remember, but you just install a screensaver, and this screensaver is using your computational resources while you are not using your PC. The other guy from the internet, they use your computational powers to solve this problem, for the protein folding problem, okay? Because the solution space is so huge, they make even use of all the computational power in the world. They try to make use of all the computational power in the world to solve this problem. There are supercomputing centers around the world that tries to do that because it's really computationally really expensive to exhaustively search the whole solution space. And there are three main approaches to this problem. I'm going to talk about those in this class, and we are not going to go into much detail about these algorithms. I'm just going to talk about the three main approaches. What are their differences? How do they solve the structure at a high level? If you want to learn details about these algorithms, you're not going to learn in this class is detailed enough to implement them actually. So you're not going to have a homework to predict protein structure, and that's going to be the hardest homework of all. I mean, if I want you to devise a new technique to find the structure, so relax, you're not going to be implementing these. I mean, even the existing ones, they are so complicated, I shouldn't give them as homework, so you're not going to be working on this in your fourth assignment. But we are going to see in general how this problem can be solved. And this was the same example I gave you last hour about the complexity of the solution space. Consider as 100 residue protein. If each residue can take only 3 positions, or if you only have three options to have between the angle between 2 residues, it's going to there are this many possible conformations, okay? It's like 5 and 47 zeros after that 5. Okay, it's this many different. Of course, not all of these conformations are going to be possible because by making these turns, some conformation is going to have collisions in them, meaning that an amino acid occupies the same space as another amino acid, which is not possible. I mean, 2 amino acids may not occupy the same space. I mean, some of these angles will give you infeasible structures, but still, if you just look at the number of feasible structures, it's going to be close to this number. And if we have imagine we have an algorithm which searches this search space exhaustively. And after search, and it takes, for example, just this many seconds, 10 to the minus 13 seconds, to convert from one structure to another. And an exhaustive search would take this many years, 10 with 27 zeros. So it's impossible to do an exhaustive search, just simply saying this. And in nature, actually, I mean, the cell itself, this folding process happens really quickly. And there's research which tries to find out this folding pathway. This is called folding pathway. How does the protein sequence folds onto this three-dimensional shape in the cell? Really, how does this happen naturally? We have these sequence of amino acids. How does this sequence of amino acids folds onto a three-dimensional shape? What are the processes that are involved in this folding process? Researching this is called the folding pathways. If you are interested in this, you can also look at, there are lots of papers about this in literature. And there are certain forces that are believed to drive this folding process. So given a sequence of amino acids, usually some amino acids, they like to be close to water. They like to be in contact with water. And some of them, they hate to be in contact with water. So there are hydrophobic and hydrophilic amino acids. Hydrophobic amino acids are the ones that, so hydrophobia, so it's afraid water, so they don't want to be near water. Hydrophilic, they love to be near water. And these are, so the hydrophobic residues, I mean, they don't want to be near water. And when the protein is, the amino acid sequence is formed, they are in close contact with water molecules around it. So a protein molecule in cytoplasm, there are lots of water molecules around. So what they want to do is these hydrophobic amino acids, they tend to stay close to each other and they form a hydrophobic core, and the other amino acids, they surround them, so they prevent water molecules to interact with these hydrophobic ones. So this is, they believe, a lot of researchers believe that these physiochemical properties are the driving forces that is going to determine the final structure. And also, during this folding process, the folding occurs in such a way that inside the molecule, we do not have cavities that are void. So the minimum, the folding should probably give us a minimum volume structure. And also, there are certain amino acids that are very important. The cysteine amino acid, which contains the sulfur atom. These two sulfur atoms, they form disulfide bonds, okay, which stabilizes the structure. They are very strong bonds. They are called bridges, actually, disulfide bridges. These bridges really stabilize the structure. So as you can see, in order to solve this problem from sequence, given a sequence, what the structure is going to be, you need to know a lot of physics and chemistry. Okay, this is, this shows a computer scientist by itself. I mean, all this information, which atoms interact with each other, what are the external forces. I mean, this is really outside the field of computer science. But lots of, so maybe, but other physicists and other natural scientists, they also need computational power to solve this problem. So it should be really a work of interdisciplinary work to solve this problem. And another important thing is, if we have a structure, a protein sequence, even you change a single amino acid, you take that amino acid, you replace it with another amino acid. This simple change, a single amino acid change in a very large protein sequence, may completely change your overall structure. So, I mean, this tells you that changes are not local. That's what makes the problem difficult. By just looking at local regions, by finding out the local shapes, you cannot predict the overall structure. Even a single local change can change the complete protein structure. And this is this disease So it was sickle cell anemia, I guess. It was this disease. The mutation of this is glutamine changed to valine, okay? What happens is that, this happens in hemoglobin. It's a disease of mutation in the hemoglobin protein. So it's in the red blood cells. This is responsible for binding oxygen, so that carries oxygen. A single mutation from this amino acid to this amino acid in the beta chain. So this mutation replaces a charged glutamine by a hydrophobic valine, meaning that on the surface we had an amino acid which loved to be near water. Okay, I want to be in the surface. Okay, I'm happy to be at the surface, and this mutation takes this amino acid that wants to be near water and replaces it with another amino acid which doesn't want to be near water, hates water. Okay, so what happens is that this hemoglobin it forms imagine you have two beta chains. Okay. And there's this valine which doesn't want to be near water in both of these chains. So what happens is that they want to stick to each other to be away from water, just come together, let's be away from water. They form a sticky patch. That part of the surface becomes sticky, meaning that it's really wanting to bind to other amino acids to be away from water. So this hemoglobin, therefore, this sticky patch causes hemoglobin to stick together. So hemoglobin proteins cannot perform their functions separately. Lots of hemoglobin proteins stick to each other. They form fibers which deform the red blood cell and do not carry oxygen efficiently. Okay, the single mutation, and it was one of the first. It was the first molecular disease. This mutation was found. They identified this mutation was the cause of this disease. Also sickle cell. So the red blood cells becomes like this. Okay, their shape becomes like this. Okay, this single mutation that is shown here in the beta chain. Since there are two identical beta chains, this is the mutation at the 6th amino acid of this beta chain. Now, I'm going to, let's actually stop here. Next week, I'm going to talk about these three major different techniques. Ab initio techniques, homology modeling techniques, and protein threading techniques to solve this problem. Given a sequence, how can we find the structure of a protein?